Hello everyone, in this video we're going to talk about gene mutations, and I'm really aiming this video for those in AS and A2 at A level and IB, because we're going into quite some detail about the different types of gene mutation that you have and the causes and consequences of them. But we'll also talk about the genetic control of cell division and what happens when you have a mutation in the genes that control cell division as a process. So let's just start first of all with what gene mutations are, because that's really key. There we go. That's what they are. So I'll just very quickly, just to save time, put a little definition there. A gene mutation is a change to one or more bases or any arrangement of the bases in a gene. So I'm thinking of the letters, I'm just very roughly drawing this. So if we imagine drawing A, C, G, let's put in a T, and let's just, for the sake of argument, have another G. So here is a strand of DNA with our nitrogenous bases, adenine, cytosine, guanine, thymine, and then another guanine. If we change one or more of the bases or arrangement of the bases, that is a gene mutation. Now that may result in a change in the amino acid sequence of the polypeptide that we make when we transcribe and translate that gene. So that's really key to note. Any of these mutations can ultimately lead to a change in the amino acid sequence of the polypeptide that results, and that's really key to note. So let's think about some of these gene mutations, types of gene mutation. So the first one to consider is the addition mutation. Now, as the name suggests, there we add an extra nucleotide to this base sequence. So it's where we have an extra nucleotide inserted. So an extra base is added to the sequence. So a nucleotide is a part of the sugar phosphate backbone. So this part of the strand, part of this strand that I've drawn here, and the base. So a sugar, a phosphate, and one of the bases is what we call a nucleotide. So by adding an extra nucleotide, so addition, we add one nucleotide, then we're putting an extra base in the sequence. So we could get something like A, C, G, and then randomly an extra A appears, then T, and then we have G. Now, if you've learned about the triplet code, you know that the base sequence is read as a sequence of 3, a codon. So at the top, we would read this as ACG, then TG, and whatever the next base in the sequence would be here. But by adding this extra A, you can see we've changed the order that the sequence is read in, because we'll read this now as ACG, ATG. So by reading that as ATG, that's going to alter the amino acid that would essentially appear in our polypeptide chain that we make when we transcribe and translate this particular base sequence. So the first type of mutation is an addition. The second type that we need to cover is a substitution mutation, and that's when we replace a nucleotide with a completely different base. And there are 3 possible consequences of a substitution. So there are almost 3 things to consider here, 3 eventualities. We could get what's called a missense mutation. Now in a missense mutation, that's when the base change simply results in a different amino acid being coded for. So instead of ACGTG, we may have ACCTG. We change the G to a C. We've substituted that particular base, so we make a different amino acid. We could end up with something called a nonsense mutation. And in a nonsense mutation, that's when the base change results in the formation of one of the 3 stop codons. Now in another video I talked about a stop codon as being a sequence that stops or terminates translation. So when we're transcribing, or when we've transcribed messenger RNA from our gene, when we're going to synthesize a protein, and then we translate that messenger RNA, we form a polypeptide chain, but with a stop codon, we terminate that chain, if you like. So this mutation might prematurely terminate that chain that we're forming. And the final one to note, really important type of mutation, is a silent mutation. Now when we have a silent mutation, that's when the substituted base, although it's different, still codes for the same amino acid. There are certain amino acids that have multiple base sequence codes for them, and that's what happens in a silent mutation. The base changes, but we end up just making the same thing. So let's think about some of the other types of mutations. So there's addition, substitution. For this, I'm just going to shrink the screen just a little bit to give us a little bit more room. If I'll move this over to here. So number 3, let's think of another type. We've got deletion. Now as the name suggests, that's where a nucleotide is completely lost from the normal DNA sequence. So where we had ACGTG, and just to save time, I'll write this all in the same colour. So where originally we had ACGTG, that may become ACTG. We've completely deleted, if you like, that G. So you can see again, when we read it as a set of 3 bases, ACT is very different to ACG. So when we have a deletion mutation, a whole nucleotide is lost from the normal DNA. We could have something called an inversion mutation. Now when we have an inversion mutation, that's when the sequence literally is inverted. So again, we'll go back. We'll use just a different colour this time again. So that's when ACGTG could very easily, if that portion gets inverted, could become GTGCA. So you can see that sequence, that small sequence there is reversed. So once again, when read as a series of 3 bases, a triplet code, we can see it's a completely different order that we have there. And one last one to mention is duplication. So I'm not going to write that down because the main ones are the addition, substitution, and deletion, but we can get duplication. So we may get ACGTG, ACTTG immediately after. So we get a second copy of the whole of the sequence. So even though there'll be hundreds, thousands perhaps of bases in one particular gene, this small section of bases might duplicate. So you can see in all these examples, we've had a change to one or more bases or the arrangement of bases in the gene. And that by definition is what a gene mutation is. So let's think about some of the consequences. So I've outlined at the top here one of the key consequences in that you can end up completely altering the polypeptide that gene codes for. Now that could cause the cell a number of problems. It could prevent cell growth, it could cause the cell to produce the wrong product or the wrong protein. Now, if that's an enzyme, for example, that the cell was meant to make, then you'll make a non-functioning enzyme. So the process that enzyme is part of no longer operates. If that protein, for example, is used as a receptor, then you no longer have that function available to you. So the consequences can be devastating. One of the main consequences of gene mutation may be cell proliferation. It may cause abnormal proliferation and growth, which we're going to come on to in a moment. The causes are really important, the causes of these gene mutations. That's something we need to consider. Let's shrink this down a little, because it's really important that we consider the causes of these gene mutations. Now, one thing to add is that they can be spontaneous. That's really important to know, that you can just get spontaneous mutations. Especially during DNA replication, it's quite random. You normally get about 1 to 2 mutations per 100,000 genes per generation. But the rate of gene mutations can be increased by what are called mutagenic agents. Now, mutagenic agents basically means anything that could act as a mutagen, something that's going to mutate this DNA. So we're thinking maybe high energy radiation, but also chemicals. Now, high energy radiation can disrupt the DNA molecule, but also chemicals can alter the DNA structure and can even interfere with transcription. So there's 2 main causes of gene mutations. What we're going to look at in this final part of the video is the genetic control of cell division. And I think I'm going to use a separate page to talk about this. So we'll use a separate page here. And now we're going to look at what's called the genetic control of cell division. That's where we're going to end this video. Now, cell division is controlled by genes, and it's controlled by 2 particular types of genes. The first one that I want to mention are the Proto-oncogenes. Now, the Proto-oncogenes are the ones that stimulate cell division. So proto-oncogenes stimulate cell division. So you might be able to guess that the second one that I'm going to mention are the ones that slow cell division down. And these are known as the Tumour suppressor. Apologies, 1P. Tumour suppressor genes. And they are the ones that slow cell division. So let's look at how these actually interact with one another. So initially we have, I'll just write down here, an inactive, and I'm just going to call it PO for the moment, proto-oncogene. So we have an inactive proto-oncogene. Now we have something called growth factors. I'm just going to put in here as a separate thing to mention. So we have growth factors, and growth factors are chemicals that attach to receptors on the cell membrane of normal cells via what are called relay proteins, and they switch on or activate genes that stimulate DNA replication. So with those growth factors, what we're going to do is get an active proto-oncogene. So that's going to stimulate cell division. So I'm just going to draw one cell here. In fact, I'll draw a set of cells with nuclei here. And when those proto-oncogenes are active, we stimulate cell division. Now, that would lead to rapid cell growth. So clearly you wouldn't want that, because by definition, uncontrolled proliferation of cells can actually lead to tumours and cancers. So we have normally our tumour suppressor gene. So I'm just going to call this TSG for the moment, our tumour suppressor gene. And our tumour suppressor gene actually inhibits this proto-oncogene to a degree to give normal cell division and cell growth. So when we inhibit the active proto-oncogene, the tumour suppressor gene is able to control cell division, slow it down, and we have normal cell growth. Now that's normally what happens when we have cell division, and these are the genes that are involved in controlling it. But this video is all about mutation, so let's think about what happens when you have a particular gene mutation. So let's start with the proto-oncogene. If you have a mutation in the proto-oncogene, that could turn that proto-oncogene into just a straight oncogene. So it loses that word proto, if you like. So the proto-oncogene can become an oncogene if it mutates. Now that can have quite a devastating consequence. If that proto-oncogene mutates into an oncogene, then you could have that receptor on the cell membrane, the relay protein, if you like, that I mentioned, being permanently stimulated. Now that would cause cell division at an increased rate. Or you may actually, from an oncogene, get an excessive amount of growth factor produced. And that too would stimulate cell division beyond the normal. Now let's think about the tumour suppressor gene. If we actually had a mutation in that tumour suppressor gene, and that tumour suppressor gene no longer functioned, so we'll put a line through it, if that no longer functioned, we would lose that inhibition of the proto-oncogene. So we would stimulate cell division and it would be uninhibited. There'd be nothing to regulate its rate. So the cells will grow and divide, and mutant cells can actually form. Now they could clone and form tumours. Now those tumours, I have done a separate video on tumours, and those tumours could be benign or they could be malignant, and it's the malignant ones that are the cancerous ones. So if we have a gene mutation of any of the types I've mentioned in this video, of that tumour suppressor gene, we have nothing to inhibit the proto-oncogene, so we get stimulated cell division. The proto-oncogene and tumour suppressor gene almost regulate together the rate at which cell growth and division occurs, and a mutation in either we can see can seriously affect that. Okay, hope all of that helps.